Hi, we meet again today for lesson 6. For today's lesson 6.3 light independent reaction. Okay, so today topic light independent reaction, or we say dark reaction. Why? Because light independent reaction means that the process doesn't need a light, so it happens somehow at night or day. It just happens when the ATP and NADPH required enough for them to do a process. Okay, what is dark reaction? Actually, dark reaction or Kelvin cycle is the name of the process. Is the second stage of photosynthesis that follows light reaction. Remember that in the light reaction produce ATP and NADPH. Now that reaction uses this ATP and NADPH which to form a glucose or food. Okay, so previously I already shown you that plant is a powerful factory that can produce food for us or for any life in the earth, because in the that reaction or Kelvin cycle they will produce sugar or food. Okay, so in this stage, carbon dioxide. So light reaction use water and light to produce ATP and NADPH, while in the dark reaction, carbon dioxide were used to convert into sugar. So that reaction takes place in the stroma of chloroplast. Okay, so the stroma of the chloroplast is the site of the Kelvin cycle. So here, the powerful factories exist. Okay, so now, although it takes place during the day, this reaction is not dependent on light. So as I said, it happen in certain plants. It happen during the day, and although it happened during the day, this reaction not dependent on light. The formation of sugar is a cyclic process of enzyme catalyzed reaction. So, in this lesson, I want you to explain the mechanism of carbon cycle. Later, after finish the class, compare C3, C4, and CAM plants. Okay, differentiate the anatomical and physiological variations between C3 and C4 plants. Role of G3P in the formation of carbohydrate, protein, and lipids. So today we only discuss and learn about mechanism of carbon cycle and role of G3P in the formation of carbohydrate, protein, and lipids. So we start with Calvin cycle. Okay, this is Calvin cycle. It's a sugar producing process that occurs in mesophyll cells of C3 plants. It consists of three stages, namely carbon dioxide fixation, reduction, and regeneration. So I click play so you can observe the stage 1 of the Calvin cycle. So if you notice from here, carbon enters the Calvin cycle in the form of carbon dioxide and produces G3P, which is also called PGAL. So now if you notice there, mention that three molecules of carbon dioxide. Okay, so actually for the cycle, only one carbon dioxide molecule is incorporated at a time, but the cycle must be completed three times to produce a single 3 carbon G3P molecule. Okay, to produce a single 3 carbon G3P molecule. So later you will notice that 6 times to produce a 1 carbon, sorry, 1 carbon, 1 glucose molecule. I want to say that 6 carbon glucose molecules equal to 1 glucose molecule. Okay, I just continue first. So stage 1 in the Calvin cycle is a carbon dioxide fixation. Okay, how carbon dioxide fix into the receiver of carbon dioxide is a ribulose bis phosphate, RuBP. RuBP uh, is in the form of 5 compounds carbon. So here RuBP combines with carbon dioxide. All right, but here mention 3 molecules ribulose biphosphate because in in order to make one G3P, they need 3 carbon molecules, but only one carbon dioxide molecule is incorporated at one time. So at one time, one carbon dioxide incorporated with ribulose bisphosphate. Okay, because of 3 carbon dioxide enter the Calvin cycle in turn. So here mentioned 3 molecules ribulose bisphosphate RuBP 5 carbon compound. Okay, to form. Okay, so one RuBP incorporated with 1 carbon dioxide. So 5 carbon compound plus 1 carbon dioxide. All right, so it will become 6. Sorry, 1, 2, 3, 4, 5, 6 unstable 6 carbon compound. So then immediately converted into 3 phosphoglycerate, 3PG. One 3PG molecules is a 3 carbon compounds, if you can see at here. How many carbon molecules fixed with RuBP? So it is 3 molecules of carbon molecules will fix with RuBP. So what is the enzyme that catalyzes this reaction? If you can see there, it is rubisco, ribulose, bisphosphate, carboxylase, oxygenase. So you can call this enzyme rubisco. Yes, you can write this enzyme with this name only, rubisco. Okay, and then stage 2 of the Calvin cycle is a reduction stage. Okay. Now, if you can see, 3 PG molecules bonds with phosphate group and is converted into 1,3 bisphosphoglycerate. If you notice here, previously 3 carbon compound, 1, 2, 3. Okay, because previously I already explained about 3 carbon dioxide, so it becomes 6 molecules of 3 PG. We receive 6 ATP to become 6 ADP. Okay, because it is a reduction process. If you notice here, there only one phosphate group at the 3PG, then when it receives energy in the form of ATP, so another phosphate group attached to the 1,3 bisphosphoglycerate. So that's why the name is 1,3 bisphosphoglycerate. We continue. Now, the phosphate group required for this reaction comes from ATP produced in light reaction previously. So how many of molecules of ATP are utilized here? If you can see, it is 6 molecules of ATP needed. If 3 carbon dioxide molecules enter the Calvin cycle to produce 1 G3P. Okay, so now I click on reduction of 1,3-BPG. So in the reduction process, 6 NADPH reduced to NADP plus by releasing 6 PI. So it is reduced to G3P by the release of 6 PI. So right here, we continue. G3P is formed by accepting a pair of electrons from NADPH produced in light reaction. So the removal of phosphate group replaced by hydrogen atom. Okay, now how many molecules of NADPH are utilized here? It's also 6 molecules of NADPH being utilized in this reduction stage. How many molecules of G3P are produced? It is 6 molecules of G3P are produced. So among the 6 molecules of G3P, remember this please, 6 molecules G3P produce, one molecule will exits the cycle and the remaining 5 molecules of G3P will enter the next step of the cycle. So here the molecule that exits the cycle is utilized in producing sugars, proteins and lipids. Okay, then in the stage 3 of the Calvin cycle is a regeneration process. Okay, so during regeneration process, G3P combine with the phosphate group and form 3 molecules of 5 carbon RuBP again. Okay, so remember 5 times 1, 2, 3. It is 15 carbon compounds. Okay, then it will produce 3 RuBP. RuBP 1, 2, 3, 4, 5 carbon compounds. So from 5, they will rearrange into 3 molecules RuBP 5 carbon compound. Okay, so from 5, G3P combine with the phosphate group and will form 3 molecules of 5 carbon RuBP. So where does the phosphate group comes from? From ATP. So how many molecules of ATP are utilized here? So from 5 molecules, they need to form 3 molecules. So only 3 molecules of ATP are utilized here. So for overall, you need to observe the carbon cycle again in the stage of carbon fixation, 3 carbon oxide, sorry, 3 carbon dioxide enter the carbon cycle to produce 1 G3P. In the process of in the stage of reduction, they utilize the 6 ATP, and later on the regeneration phase, G3P utilize 3 ATP to rearrange and become 3 molecules of ribulose bisphosphate RuBP. And then how many molecules of ATP utilized for 1 Kelvin cycle? So for 1 Kelvin cycle, it is 9 molecules of ATP, 6 ATP at the reduction phase and another 3 at the regeneration phase. Now, how many molecules of NADPH oxidize on these 6 molecules at the reduction stage? So C3 plants are those plants in which the first product formed after carbon fixation is a 3 carbon compound. So if you notice, the first product formed after carbon fixation is 3PG. So for those plants running this carbon cycle, we call C3 plants. Okay, so what is the significance of carbon cycle? It fixes carbon dioxide. It's amazing, right? From carbon dioxide from the atmosphere and results in the formation of sugar as a food for the all the living things. All right, so okay, so do you remember the Rubisco? It is enzyme for the carbon fixation. So Rubisco catalyzes the carbon fixation of carbon cycle in C3 plants and produces a 3 carbon sugar. Okay, so I continue. What happened to the if you notice, one J3P will exit the cycle, right? So what happened to the fate of G3P that exit the cycle? So G3P produced in Calvin cycle previously is used for the synthesis of all other carbon containing substances such as sugar, okay, proteins, and lipids in a green plant. Okay, let's see how sugar was formed. Okay, a large proportion of G3P is converted into sugars. Okay, somehow phosphorylated into six carbon sugars, glucose, disaccharides, example sucrose, polysaccharide sugar, example starch and cellulose. Okay, so if you can observe here, G3P is converted into 6-carbon phosphorylated sugars. The phosphorylated sugars further converted into sugars like I mentioned, okay, sucrose, starch, and cellulose. Then the sugars may be used in respiration for the plant itself to provide ATP for the plant cells. So they are also used for storage and growth. Okay, for storage in the plants, it is our main source for the foods. All right, now I click on products again. Okay, how about proteins? Sorry, how about lipids for the plants itself? Okay, first, how are fatty acids produced? Okay, G3P is converted into acetyl coenzyme A, then further converted into fatty acids. Glycerol is formed directly from G3P itself and combined with fatty acids to form lipids, triglycerides, and phospholipids. So these lipids are utilized for cell membranes and storage. And then I click on proteins. Okay, now G3P is converted into acetyl coenzyme A, which enters Krebs cycle. Then in the Krebs cycle is converted into alpha ketoglutarate by a series of reactions. Then alpha ketoglutarate is converted into glutamate. Then glutamate goes through a process called transamination in which amino acids and proteins are produced. So plants use these molecules, protein and amino acids for the production of nitrogen containing compounds such as DNA and RNA nucleotides. Okay, so for G3P, produced in the carbon cycle is used for the synthesis of sugars, amino acids, and fatty acids. Sugars formed are glucose, sucrose, starch, and cellulose. Lipids are formed from glycerol and fatty acids. Glycerol formed directly from G3P, and the fatty acids are built from acetyl coenzyme A. Proteins are formed by the conversion of G3P into acetyl-CoA, which enters Krebs cycle where it is converted into alpha-ketoglutarate, which is further converted into glutamate. Glutamate undergoes transamination to form amino acids and proteins. Okay, so for today, that's all. As I mentioned previously, for today, you just need to know or you will explain the mechanism of Calvin cycle and describe the role of G3P in the formation of carbohydrate, protein, and lipid. And the significance of the Calvin cycle, remember that only one carbon dioxide molecule is incorporated at a time. So the cycle must be completed 3 times to produce a single 3-carbon G3P molecule and 6 times to produce 6 carbon glucose molecule. So, summary of the Calvin cycle is production of one molecule of G3P run through one Kelvin cycle. Okay, so the one molecule of G3P will require 3 molecules of carbon dioxide. So the cycle will fix the carbon dioxide 3 times. So 3 times carbon fixation with RuBP require 9 ATP and 6 NADPH for one Kelvin cycle. However, for net gain of one molecule of glucose, the plant need 2 molecules of G3P. So the plants need a 2 Kelvin cycle to form one molecule of glucose or food. So total the 2 Kelvin cycles require 18 ATP and 12 NADPH. It's just a simple math. You just need to understand 1 Kelvin cycle. For the 1 Kelvin cycle, then to fulfill the food production, the 2 Kelvin cycle needed to form one molecule of glucose. Okay, I hope you can summarize this all. Okay, understand this. And later, please do the worksheet that I gave to you, and we will see again in the next lesson. Okay, that's all for today. Okay, bye.